All right, today we're going to talk about muscles. There are a couple different chapters we do on muscles. This one's going to focus more on their function. So when we look at muscular tissue, there are three types of muscle tissue. Your skeletal muscle tissue is called this because most of the skeletal muscles move bones. It's striated in its appearance, which means it's going to look like it's striped. There's light and dark bands when you look at it underneath the microscope. It's under voluntary control, so there's going to be conscious control of this. You're also going to have conscious awareness of it when you do control it. Some of the skeletal muscles are also controlled subconsciously to an extent. So if you look at your diaphragm, for example, it's going to alternately contract and relax without conscious control. However, you can consciously override that as well. Cardiac muscle is found only in the walls of the heart. It's also striated in its appearance, but it's under involuntary control. So your heart is going to know to contract regularly without you consciously remembering for that to happen. That's a good thing. So the heart is going to have its contraction initiated by the pacemaker. And then smooth muscle is in the walls of hollow organs. This would also be your blood vessels, airways, as well as some organs like the uterus and stomach. It does not have striations that you see in the skeletal and cardiac muscle, which is why we call it smooth. Instead, they're spindle-shaped with tapered ends, and this is usually involuntary. So this here just gives a comparison in these different fibers, the different appearances of them, the different sizes and strength of them, and then how well they can regenerate, which you can see on here. The skeletal and cardiac muscle have limited abilities to regenerate. It's a little bit better with the smooth muscle depending on where it is. So muscular tissue is used to produce body movements. So this can be walking, running, moving the arms. It helps to stabilize body position, so it's going to help hold your posture. It's going to move substances within the body. So we do this in the digestive tract through peristalsis. And then your heart is going to move stuff as it pumps blood. It will also pump the nutrients through your body. And then it generates heat. So when a muscle contracts, one of the byproducts that is produced as a result of doing that metabolism is heat. This is what allows us to stay warmer so we don't have to stay in an environment that's 98.6. The whole reason you shiver is to try and increase heat production. Muscle tissue is going to have excitability, contractility, extensibility, and elasticity. So excitability means it's going to respond to stimuli. It will contract forcefully when it is stimulated. It has the ability to stretch out without being damaged. And then it has elasticity that will allow it to return to its original length. So when we look at the connective tissue components, the fascia is going to be the dense sheets or broad bands of irregular connective tissue that surrounds muscles. It will tend to surround groups of muscles. The fascia actually has connections that go throughout your entire body. Epimysium is the outermost layer. It's going to separate between 10 and 100 muscle fibers into bundles called fascicles. So then when you make the fascicles, they're going to be surrounded by perimysium. And then the endomysium is going to wrap around the individual muscle fibers. It will also contain blood vessels, nerves, and satellite cells, which are your embryonic stem cells that help function in the repair of the muscle tissue. So the collagen fibers of all three layers are going to come together at the end of the muscle. And when it comes together, where now these are sitting on top of each other, it's going to form a tendon or an aponeurosis. So tendons are just cords that will attach a muscle to a bone, where an aponeurosis is more of a broad, flat tendon. So skeletal muscle is going to be composed of muscle cells. We call them fibers. You'll have connective tissue, blood vessels, and nerves. The fibers are long, they're cylindrical, and multinucleated. So they tend to be smaller in diameter in small muscles and larger in diameter in the large muscles. So they can range from anywhere from 1 millimeter to 4 centimeters in length. So muscle tissue developed from myoblasts, and the numbers of muscle cells will remain constant. Your skeletal muscle is striated in appearance, and you'll notice that the nuclei are peripherally located. So this is showing here a cross-section. So you can see where you've got these different fascicles that are going to have perimysium around them. You can see the individual muscle cells, and when all of this continues on, now you've got these connective tissues layers on top of each other, so it forms the tendon. Then it's actually going to split and be a continuous tissue that will cover the bone to make the periosteum. This is part of what makes their attachment onto the bone so strong. So when you look at the nerve and blood supply, your somatic motor neurons, which somatic means to the body, motor is going from the brain down to the body, it's going to stimulate muscle fibers to contract. The neuron axons are going to branch so that each muscle cell or fiber is going to be innervated, and it's going to form a neuromuscular junction. Sometimes it's called a myoneural junction. There'll be capillary beds that will surround the muscle fibers because muscles require large amounts of energy. This is going to give them more energy from the vascular network to deliver oxygen to them. It will also deliver the nutrients and carry away the metabolic waste. The number of skeletal muscle fibers is set before you are born, so most of these cells are going to last a lifetime, but they can grow. So muscles grow by hypertrophy, which would be enlarging the existing muscle fibers. Testosterone and human growth hormone are both hormones that will stimulate hypertrophy. This is why it's so much easier for males to build muscle than a woman. For a woman, she's not accidentally going to build muscles that are going to look like a bodybuilder. Your satellite cells, or myosatellites, are going to retain the capacity to regenerate damaged muscle fibers. The muscle nuclei are added in strength training and not lost, which is one of the reasons why you can say that there is a possible muscle memory in there. So once you make those nuclei, they're still there. The muscle may actually decrease in size, but the nuclei are still there. So the sarcolemma, this is going to be the plasma membrane of a muscle cell. Let's go back. The transverse or T-tubules, these are tunnels that are going to occur in the plasma membrane. This is going to allow the action potentials to travel through the T-tubules from one cell to the next. The sarcoplasm is the cytoplasm in a muscle cell fiber. It's going to include glycogen that's used to make ATP and myoglobin, which is going to be the red-colored protein that will bind and carry oxygen molecules. Myoglobin will release oxygen when it's needed for ATP production. So your myofibrils are thread-like structures that are going to have a contractile function. The sarcoplasmic reticulum is similar to the endoplasmic reticulum. It's going to be membranous sacs that are going to encircle the myofibrils. These are going to store calcium ions, and they will release calcium ions to help trigger muscle contraction. So the filaments are going to help to function in the contractile process. We've got thick and thin filaments. There are 2 thin filaments for every thick filament. And these are going to be rearranged in these units, the basic functional unit of a myofibril, which will be the sarcomeres. Those are just the compartments of the arranged filaments. So you can look on this picture, and here you can see this is a sarcomere. So you would have another sarcomere here, another one here, and so on. So there's these repeating units. This is looking at your muscle here. This is looking at a fascicle. Here we go in and look at having thin filaments and thick filaments. And this is looking at that a little bit larger. Again, you can see in the myofibril here that you've got thick and thin filaments. So with the skeletal muscle tissue, we have a model that looks at these different bands and zones. And this can be helpful for some people in understanding how the muscle contracts. For other people, I feel like this model just makes things more confusing. So if you're in that boat, don't get too worked up over it. You can still understand how the muscles function, and then the model usually comes together afterwards. So the Z-discs are going to separate one sarcomere from the next. So if we go back to look at our picture of a sarcomere here, you have a Z-disc here, and you would have another Z-disc here that's going to separate the sarcomeres. The A-band is the dark part of the middle of the sarcomere. Here you're going to have thick and thin filaments overlap. Where the I-band is lighter, it's going to contain thin filaments only, no thick filaments. So the Z-disc is going to pass through the center of each I-band. So if we come back and look here, you can see the Z-disc and the Z-disc here passing through the I-band. The H-zone is in the center of each A-band. It's going to have thick fibers but no thin fibers. The M-line, this is going to contain supporting proteins that are going to help to hold the thick filaments together in the H-zone. So you're going to have this zone of overlap where you're going to have thick and thin filaments overlapping each other. So here you can see there's the M-line here in the center. You've got this A-band here, the I-band on the sides, and then this H-zone in the middle, the M-line down the very center of that. And then this is showing it here again, Z-discs, the I-band, A-band, H-zone, and M-line down the middle. And this again is going to show where you go from the skeletal muscle, having your fascicles, the individual muscle fiber, the myofibril, and the arrangement of the sarcomere within the myofibril. So when we look at the contraction and relaxation, you'll notice here that these fibers are partially overlapping. What's going to change when the muscle is contracted is you're going to have more overlap of the fibers. They're going to slide past each other. None of the fibers actually shorten. So our contractile proteins are going to be the ones that generate the force during contraction. We have myosin, which is a thick filament. It acts like a motor protein. So it will convert ATP to the energy of motion and pull on the actin. So each of those myosin molecules will protrude outward with a myosin head. Actin is composed of G-actin and nebulin. This is going to make up one of the thin filaments. They're going to provide a site where the myosin head can attach or grab on. Tropomyosin and troponin are also thin filaments. In relaxed muscle, myosin is blocked from binding to the actin because you're going to have strands of tropomyosin cover the myosin binding sites. So the calcium ion is going to bind to the troponin to move the tropomyosin away from the myosin binding sites so they become exposed and myosin can bind to the actin and the muscle contraction can begin. So we do have some regulatory proteins that will switch the contraction process on and off. So the tropomyosin and troponin are big for this in allowing that to happen. So structural proteins, these will align the thick and thin filaments properly. They're going to provide elasticity and extensibility, and they'll link the myofibrils to the sarcolemma. So the first of these is titin. It's going to stabilize the position of the myosin. It will function in elasticity and extensibility of the myofibrils. Dystrophin, this is going to link the thin filaments to the sarcolemma. Myomesin is going to form the M-line. That holds the thick filaments together. And then the alpha-actinin and connectins are going to make up the Z-discs. So when we look at muscle contraction, we say it's a sliding filament mechanism that will explain the relationship of the thick and thin filaments as the contraction proceeds. It's a cyclic process, so it's going to begin with calcium being released from the sarcoplasmic reticulum. That binds the troponin. Troponin is going to move the tropomyosin. to expose the myosin binding sites on the actin. Myosin will grab onto the actin, forming what we call a cross bridge, and it will bend towards the H zone. So ATP allows the release of the cross bridge. So what happens is you can see the size of the H zone here when it's relaxed versus the size of the H zone when it's contracting. These have just slid towards the center. When you look at the I bands, those also get smaller in contracting fibers. So none of the fibers shrink, they just slide past each other to have more overlap. So this is showing here where you've got these structural proteins helping to hold things together. So here you can see this myosin grabbing onto the actin. That's what we refer to as forming the cross bridge. So this is showing the different zones and bands here underneath the microscope. So here you've got the thick filament, which is myosin. These are the myosin heads, this is the tail. And then here is the actin. You've got the troponin and tropomyosin here, and this is going to have your myosin binding sites covered. So when we look at the contraction and relaxation, the cycle has 4 steps. The first is ATP hydrolysis. So this will reorient and energize the myosin head. It's basically going to change the shape of it a little bit. It will form the cross bridge. So it's going to attach to the myosin binding site on the actin. The third phase is the power stroke. This is going to be where the cross bridge rotates and slides the filaments. So it's going to ratchet and pull on it. And then the last is the detachment of the myosin from the actin. As the next ATP binds to the myosin head detaches from the actin. So your contraction cycle will repeat as long as ATP is available and the calcium level is sufficiently high. If either of those run out, it's not going to be able to contract anymore. So continuing cycles will apply the force that will shorten the sarcomere. So here you can see the amount of overlap here in a relaxed muscle versus a partially contracted muscle versus a fully contracted muscle. So this just shows the cyclic nature of it here. So first the myosin heads are going to hydrolyze ATP and will become oriented. Here they are going to form the cross bridge where it's going to grab onto the actin. You're going to have the power stroke where it's going to pull it towards the center. So you'll notice it has changed shape slightly. And then as the myosin heads bind ATP again, the cross bridges will detach and you can repeat the cycle over again. So rigor mortis, we've seen this alongside the road with roadkill at various places, I'm sure. So after death, the calcium ions are going to start to leak out of the smooth endoplasmic reticulum. This is going to cause some of the muscle contraction cycle to start. ATP synthesis is stopped, so it's not going to have the ATP in order to reverse the process. You've got the ATP in order for it to contract, but you don't have enough for it to reverse. So these cross bridges can't break apart, and the muscles will be in a state of contraction. So depending on the source, here this source says it starts about 3 to 4 hours after death. I have read in other sources that you start to see signs after about an hour, and it lasts for about 24 hours when the cross bridges are finally broken down. So there is a length-tension relationship in the muscles. The forcefulness of a muscle contraction depends on the length of the sarcomeres. When a muscle fiber is stretched, there's less overlap between the thick and thin filaments, and the tension or forcefulness is diminished. When a muscle fiber shortens, the filaments are compressed, and fewer myosin heads make contact with the thin filaments, and tension is diminished. So you can see there is a place where it's diminished and where you've got the peak strength, and then where it's diminished again. So understretched isn't good, overstretched isn't good. There's a place where it's optimal. So when we look at the excitation-contraction coupling, if you have an increase in the calcium concentration in the muscle, it will start the contraction. A decrease in calcium will stop it. So an action potential is actually going to cause calcium to be released from the sarcoplasmic reticulum into the muscle cell. The calcium is going to move the tropomyosin away from the myosin binding sites on actin and allow the cross bridge to form. So the muscle cell membrane will contain calcium pumps to return the calcium back to the sarcoplasmic reticulum quickly so this process can repeat. Decreasing calcium ions in the sarcoplasm will eventually lead to not being able to contract. And that's part of what can happen with muscle fatigue. So as the calcium levels in the cell drop, myosin binding sites are going to be covered and the muscle will relax. So again, here is some more pictures showing this in here. The calcium channels release, and then the channels are closed. So the troponin holds the tropomyosin in position to block the myosin binding sites during relaxation. So when you open the calcium channels, calcium comes out. It's going to bind the troponin, changes the shape of the troponin and tropomyosin complex, exposes the myosin binding sites, and we have contraction. This again is showing here the sarcoplasmic reticulum with the calcium coming in and binding on to the tropomyosin. So your neuromuscular junction, this is going to be the connection between the nerve and the muscle. They have a thread-like axon that's going to extend from the brain or the spinal cord to a group of muscle fibers. So we call this the neuromuscular junction. Action potentials arise at the interface of the motor neuron and the muscle fiber. So a synapse is a site of communication. This will occur between the somatic motor neuron and a muscle fiber. We also have synapses in the nervous system. The synaptic cleft is the gap that's going to separate the two cells. So they do not technically touch each other. There is a little bit of space there. The neurotransmitter is a chemical used by the nervous system to communicate. So your initial cell communication with the second cell is going to be spread by a neurotransmitter. The synaptic vesicles are the sacs suspended within the synaptic end bulb. They're going to have molecules of the neurotransmitter acetylcholine. The motor end plate is going to be the region of the muscle cell membrane opposite of the synaptic end bulbs. It will have receptors for the acetylcholine. So here's a scanning electron microscope picture of it here. Blood vessels in here. You have the axon terminal, synaptic bulb ends connecting. And then a non-black and white picture. So a nerve impulse is going to elicit the muscle action potential. So you're going to have that action potential travel down the nerve, and when it gets down to the end of the nerve, you're not going to be able to have the action potential jump over the space. So instead, it's going to release acetylcholine. So the nerve impulse will arrive at the synaptic end bulbs and cause the vesicles to release acetylcholine into that cleft. Here you'll have the activation of the acetylcholine receptors. Acetylcholine binds the receptor on the motor end plate. That in turn is going to open an ion channel and allow the flow of sodium inside the muscle cell. This inflow of sodium is going to make the inside of the muscle fiber more positively charged, which triggers a muscle action potential. And this will propagate to the sarcoplasmic reticulum to release stored calcium. So we call this the production of the action potential. Finally, we have termination of acetylcholine. It's going to only be short-lived. It's rapidly broken down by the enzyme acetylcholine esterase. This allows for repetitive contraction of a muscle. So here you can see you've got this action potential coming down. It's going to trigger the release of these vesicles that are going to release the acetylcholine into the cleft that will bind with the motor end plate here. So these are all just different pictures that show this happening as a process. Use the ones you like, ignore the ones you don't. So here the nerve impulse is going to arrive and trigger the release of acetylcholine. It diffuses across the cleft. Acetylcholine esterase is going to follow it. It's going to break it down so it doesn't stay in the cleft and have continual contraction. So now you're going to open the calcium channels. That's going to allow the calcium to bind the troponin and expose the myosin binding sites. And now you'll have contraction with the power stroke. The calcium channels are going to close and they're going to get pumped back in using ATP. The troponin and tropomyosin complex are going to slide back into position and block the myosin binding sites, and the muscle relaxes. So part of what allows it to relax is the acetylcholine esterase will break down the acetylcholine in the cleft. The calcium levels are going to drop, and it's going to leave the myosin binding sites uncovered. After contraction, a muscle fiber will return to resting length by elastic forces, so this can come from the tendons and ligaments. Opposing muscle contraction, so if you've contracted the biceps, the triceps group can oppose that. And gravity, just bringing things back down. So this table here tries to summarize what happens. You'll have the action potential coming in. Acetylcholine's released. Sarcoplasmic reticulum releases calcium. You had the action potentials reach to the T-tubules. Your active site will be exposed. You're going to make the cross bridge. Contraction begins. Acetylcholine esterase will remove the acetylcholine. The sarcoplasmic reticulum is going to recapture the calcium. The active binding sites are no longer going to have the cross bridge, and the contraction ends. Relaxation occurs, and you can repeat the process. So this gives you a comparison of what's going to start the process versus what is going to shut it down. There are a couple of toxins that can affect this system. Botulism toxin is going to block the release of acetylcholine from the synaptic vesicles. So we don't see a lot of this anymore. It used to be found in improperly canned foods. Most of it now is actually infantile botulism that would come from giving babies honey. As an adult, you have a lot of competing bacteria, so it doesn't want to germinate in your gut, but it can in a baby. So it does not take much. A tiny amount can cause death by paralyzing the respiratory muscles. It does have medicinal uses, so Botox, so it can be used for cosmetic purposes, but it can also be used therapeutically to block certain muscles, like with strabismus, where a person has crossed eyes, blepharospasm, where they have uncontrollable blinking, spasms of the vocal cords interfering with speech. You can cause facial muscles to relax to decrease wrinkling. Sometimes they can be used for chronic neck pain due to muscle spasm, also in the lumbar region, as well as for headaches. Curare is a plant poison that has been used by Native Americans on arrows and blow dart guns. It will block the acetylcholine receptors, and it will inhibit sodium channels. So this will lead to muscle paralysis. They will actually use curare derivatives during certain surgeries to relax the skeletal muscle. Acetylcholine esterase, this is going to slow down the actions of acetylcholine and help to remove it. It can strengthen weak muscle contractions. So neostigmine is a treatment for myasthenia gravis. It's an antidote for curare poisoning, and it can terminate the effects of curare after surgery. So where do muscles get this ATP from? A huge amount of it is needed to power the contraction cycle and pump calcium into the sarcoplasmic reticulum. So the ATP inside the muscle fibers will power contraction for only a few seconds. It's going to have to be produced by the muscle fiber after the reserves are used. So there's 3 ways it can do this. 1 is creatine phosphate. 2 is by doing anaerobic cellular respiration. And the 3rd is doing aerobic cellular respiration. So what happens with creatine phosphate is it will release a phosphate onto ADP, charges it back up to ATP, and becomes creatine, which can have ATP recharge it back up to creatine phosphate. So your glycogen and glucose from the bloodstream, it can be broken down to give you glucose to go into the blood. And then you can have oxygen from the hemoglobin in the blood or from the myoglobin help to make sure that you can do cellular respiration in the aerobic mode. So with creatine phosphate, the excess ATP is used to synthesize creatine phosphate. It's an energy-rich molecule. It's going to transfer its high-energy phosphate group from ADP, which is adenosine diphosphate, to regenerate it into a new ATP. Creatine phosphate and ATP provide enough energy for contraction for about 15 seconds. This is going to depend on the intensity of the exercise. With anaerobic respiration, you're going to undergo a series of ATP-producing reactions that do not require oxygen. So glucose will be used to generate ATP whenever oxygen is available and creatine phosphate is depleted. So glucose is derived from the blood and from the glycogen stores in the muscle fibers. So glycolysis will break down the glucose into molecules of pyruvate and to produce 2 molecules of ATP. If insufficient oxygen is present, the pyruvic acid that you get from glycolysis will enter into aerobic respiration, producing a large amount of ATP. So if the organism's levels are low, anaerobic reactions could convert pyruvic acid to lactic acid, and that would be carried away by the blood. So anaerobic respiration can provide more than enough energy for 30 to 40 seconds of muscle activity. Again, it will depend on the intensity. So with aerobic respiration, the activity lasts longer than a half a minute, and it's going to depend on aerobic respiration. Here we're going to be looking at the pyruvic acid entering into the mitochondria and looking to see is it completely oxidized. When it is, it will regenerate ATP, carbon dioxide, water, and heat. So each molecule of glucose is going to give you about 36 molecules of ATP. Each of these will have 2 sources of oxygen, hemoglobin or myoglobin. So myoglobin and hemoglobin are oxygen-binding proteins. So aerobic respiration supplies ATP for 90% of prolonged activity, where aerobic respiration provides more than 90% of the needed ATP that lasts more than 10 minutes. So with muscle fatigue, it's the inability of your ability to make the muscle contract forcefully. So there's several things that can contribute to this. Inadequate release of calcium ions from the sarcoplasmic reticulum, depleting the creatine phosphate, insufficient oxygen levels, depleting the glycogen stores and other nutrients, building up lactic acid and ADP, your waste products, and then failure of the motor neuron to release enough acetylcholine. So after exercise, you still have some increased oxygen consumption. It's going to remain above resting level. So the idea of oxygen debt said that the added oxygen was taken in the body after exercise. So all this extra oxygen does have a use. It's used to restore the muscle cells to the resting levels. So it's going to convert lactic acid into glycogen. It's going to turn creatine phosphate into ATP, and it will replace the oxygen that's been removed from the myoglobin. So when we look at the contraction force, the tension or force of a muscle cell contraction varies greatly. When you have maximum tension force, it's dependent on the rate at which the nerve impulses arrive, how much stretch you have to give before it contracts, the nutrient and oxygen availability, and the size of the motor unit. So a motor unit is a motor neuron and all the muscle fibers it stimulates. The axon of a motor neuron branches out, and it will form neuromuscular junctions with different muscle fibers. So a neuron takes and makes contact with about 150 muscle fibers. So this will allow control of precise movements and consist of many smaller motor units. So muscles that control voice production have 2 to 3 muscle fibers per motor unit. The muscles controlling your eyes, they have 10 to 20 muscle fibers per motor unit. And then the muscles in the arm and leg have about 2,000 to 3,000 muscle fibers per motor unit. So your total strength of a contraction depends on the size of the motor units and the number that are activated. So for motor unit recruitment, you have the activation of additional motor units to accomplish an increase in contractile strength in a muscle. So the higher the recruitment, the stronger the muscle contraction. So this here is just showing you can have this mixed motor neuron come out and attaching to your tissues. So when we talk about twitch contraction, it's the brief contraction of the muscle fibers in a motor unit in response to an action potential. So a twitch can last anywhere from 20 seconds to 200 milliseconds per liter. The latent period is the brief delay between the stimulus and the muscular junction. So this action potential sweeps over the sarcolemma and the calcium, and the calcium will then be released from the sarcoplasmic reticulum. So your contraction period is going to be between 10 and 100 milliseconds. So your calcium will bind the troponin, the myosin binding sites on the actin are exposed, and the cross bridges form. So when you look at the relaxation period, it's between 10 and 100 milliseconds. Calcium gets transported into the sarcoplasmic reticulum. The myosin binding sites are covered by the tropomyosin. The myosin heads detach from the actin. So the muscle fibers move away from the eyes, have contraction periods lasting about 10 seconds. The muscle fibers that are going to move the legs towards the contraction is going to have a contraction period of about 100 seconds. So a refractory period is when a muscle fiber contracts. Afterwards, it's temporarily not going to be able to respond to another muscle fiber. So a skeletal muscle, this is going to have a refractory period of about 5 milliseconds. Cardiac muscle is about 300 milliseconds. So this shows here you've got this latent period, a contraction period, and then a relaxation period. Here, this is showing the single line here for the nucleus. And then your gastrocnemius. So we can have graded muscle responses where you're going to have variations in the degree of muscle contraction. This can be required for proper control of skeletal movement. So you can have your responses graded by changing the frequency of the stimulation and the strength. So how often and how hard do you stimulate things? So an increased frequency of stimulus, what's going to happen is the muscle won't have time to completely relax between the stimuli. So calcium release stimulates further contraction, and you can have temporal wave summation. You have further increase in the stimulus frequency, you can have unfused or incomplete tetanus. And then if the stimuli are given quickly enough, you'll get fused or complete tetany. So here you can see the time frame comparing to the muscle contraction strength. So this one here is showing high stimulation frequency used to complete tetanus. This would be incomplete tetanus. So muscles will contract more vigorously as the stimulus strength is increased. The contraction force is going to be controlled by recruitment. So you can have multiple motor unit summation. This is going to bring more and more muscle fibers into action. So your size of the motor units with larger and larger fibers end up being recruited as stimulus intensity increases. So this allows you to see here a single twitch with wave summation, unfused tetanus, and fused tetanus. So with isotonic contractions, here the muscle changes length and moves the load. These are either concentric or eccentric. A concentric contraction, the muscle shortens and does the work. So it would be flexion of the biceps brachii. Lowering would be into a push-up position. The eccentric contractions, the muscle counteracts or contracts as it lengthens. So this will cause extension of the biceps. So a small amount of muscle tension is in the muscles due to weak contraction of the motor units. So what you'll see is small groups of motor units are alternately active and inactive, constantly giving shift to the pattern. Muscle tone keeps the skeletal muscles firm. It helps to keep the head from slumping forward on the chest as well. So in isotonic contraction, the tension develops and remains constant while the muscle changes its length. Can be used for body movements or slowing and moving an object. Can be used for picking up a book off a table. With an isometric contraction, the tension is generated, but it's not enough for the object to be moved, and the muscle does not change length. So therefore, the muscles do not contract any further. They will stay in the same position. So holding a book steady is an example. So here you can see the differences on the chart. So when we look at the muscle fibers, they vary in their content of myoglobin. Red muscle fibers are going to have high myoglobin content. They're going to appear darker. So this would be things like the dark meat in chicken legs and thighs. They'll have more mitochondria and more blood vessels. White muscle fibers have a small amount of myoglobin. They appear lighter, so this would be like white meat in chicken breasts. So the muscle fibers will contract at different speeds and vary in how quickly they fatigue. So these get classified into three main types: slow oxidative, fast oxidative glycolytic, and fast glycolytic fibers. So your slow oxidative fibers, they're the smallest in diameter and the least powerful. They do appear dark red, so they're generating a lot of ATP, primarily by ATP cellular respiration. They do have a slow speed of contraction that will last anywhere from 100 to 200 milliseconds. They're very resistant to fatigue. They can give you a prolonged, sustained contraction for many hours. So they've been adapted for maintaining postural muscles as well as aerobic endurance type activities. So your fast oxidative glycolytic fibers, these are intermediate fibers. They're intermediate in diameter. They're going to have large amounts of myoglobin and some capillaries. They've got a dark red appearance. They do produce considerable ATP by aerobic cellular respiration. They have a moderately high resistance to fatigue. They generate some ATP by anaerobic glycolysis, and the speed of the contraction is faster. So here the twitch contractions last less than 100 milliseconds. So these will contribute to activities like walking and sprinting. The fast glycolytic fibers, they're the largest in diameter. They're going to give you the most powerful contractions. They're low in myoglobin. They've got relatively few blood capillaries and few mitochondria, so they will appear white in color. They're going to make their ATP mostly by glycolysis. The fibers contract strongly and quickly, but they also fatigue quickly. So these are good for intense anaerobic movements of short duration, like weightlifting or throwing a ball. So this picture here shows the fast versus the slow, the large versus small diameter. Here you can see neither of them are truly red, but one is different from the other. So this would be more like what you'd be likely to see in fibers that are good for endurance. These would be good for power.